This is our next INN symposium. We'll have another one coming up at the end of actually the first week of March as well, which I'll tell you about in a minute. I'm very delighted to have Mr. Julian presenting today. She's a relatively new faculty here at SFU. 3 years now? 2 years?
2.5.
2.5.
Yeah.
Still surviving, which is good. She's worked with stem cells, you know, talk with some collaboration. She has a very good ability to link up opportunities to use her platform to further inform different basic functions, but also clinical applications. I think it's a good skill. It's helpful for both my own thoughts, but also in general for neuroscience at SFU. She's a Tier 2 candidate chair. I don't know her title. That chair.
It's developmental origins of stem cell.
One.
Neural stem cell development was taken.
Okay. Good. So with that, I will turn the meeting.
Thanks, Randy. Thanks for the introduction and for the opportunity to speak today. Great to be able to speak to this group. A lot of colleagues that I've met but haven't actually had a chance to really formally present the work that my lab is doing. So this is a really nice opportunity. And really excited that the INN is happening and moving forward, and thankful to Randy and everyone behind it who's done the work to unite us as a really, a lot of people in the room who have done the work to unite us as neuroscience faculty here at SFU. We're a really broad group, but I think it's great that we're able to connect in this way now. And so my lab, we aim to be really collaborative. So, and I'm always happy to talk about new project ideas. So if anything I talk about today kind of connects with your research, feel free to get in touch, and happy to chat about any synergies that we might have. So my lab is really fundamentally, we're interested in understanding how the brain develops and how it's built. And so at the cell biological level, we really think a lot about neural stem cells and how they are able to make the really well-defined stem cell decisions that they need to make to build a properly functioning brain. So this image here is a cartoon of mammalian brain development, sort of depicting the major cell types and the way that they arrange themselves. And at the earliest form of the brain, we have the neural tube that is completely populated with neuroepithelial stem cells. And these stem cells are the drivers of brain development. They give rise to all of the cells that make up this fundamentally really complex tissue. So we want to understand which processes drive the decisions that these stem cells make to first self-renew and make more of themselves to populate the brain early on, and then to decide to differentiate into more specialized neural stem and progenitor cell types, then into neural cells and glial cells near the end of brain development. So we think about this in the normal brain. What are the important biological processes? And also, how do mutations that are linked to neurological diseases affect these processes? When you have these mutations, how is development disrupted, and what sort of effects does it have on the form and function of the brain that's formed? And ultimately, we want to use lessons that we learn from these investigations to find strategies to improve brain repair and develop better treatments for brain-related diseases. We're particularly interested in rare genetic disorders. So as we mentioned, we use human pluripotent stem cells as a primary model system to investigate these questions. And this is a really powerful system because we have a lot of control actually over how we treat these cells. We can expand them almost as much as we want and decide when we want to differentiate them into specific cell types in the brain. And okay, so this image here is just showing you kind of what a dish of pluripotent stem cells like. And these cells are reminiscent of pluripotent cells that you find in the blastocyst stage embryo during in vivo embryonic development. And those are the cells that give rise to every lineage, every cell lineage and every cell type in your body. So we can grow these cells indefinitely. And then just by changing the factors in the growth media that these cells are bathed in, we can differentiate them into neural stem cells. So that first represent these neuroepithelial cells that make up the neural tube. We can expand them in culture and they become cell types that sort of reflect these later stages of development. And then we can also control their differentiation into neural and glial cell types. And so we use pluripotent stem cell models of normal cells, and then those that also carry a disease-related mutation. And we get these either through CRISPR-Cas9 based engineering to introduce these mutations, or by reprogramming cells from a patient that has a genetic disease. We take somatic cells, usually it's a blood or a skin sample, that we can reprogram into these pluripotent stem cells, and then turn them into whatever cell types we desire. So as I said, we're really interested at the cell biological level. So we think a lot about how organelles impact the fate decisions that neural stem cells make. So, aberrations in organelle signaling has been linked to, traditional aging-related neurodegenerative disorders for a long time. A lot of changes in mitochondria, lysosome autophagy function, for example, that's implicated in those diseases. But more recently, it's becoming clear that even developmental disorders, many of them have defects in these systems. And we're finding out more about how important, organelle signaling and interactions between these systems are in development of the normal brain. So we use a variety of techniques that I'm just going to sort of brief you on before we get into the data to investigate these questions. So we do a lot of fluorescence imaging and high content imaging where we can get quantitative data from these images. And so this here, I'm showing you an example of a dish of neural stem cells. Here they are stained with not your typical antibodies even, but this is a collection of live cell probes that actually stain specific organelles. We can add these to the dish and you can visualize the staining pattern of these organelles in the cell, how much of them are there, where they're located in the cell. And we can look at these while the cells are still growing. So you can see if those patterns change, between different genotype conditions, or if you treat them with some kind of agent to perturb their function or cell fate. So they're a really, really handy tool to understand how cell behavior might be changing. And we do a lot of functional assays to sort of understand at a functional level how these cells are behaving, in a disease state, for example, as they differentiate into new cell types. So in our lab, we have a seahorse metabolic analyzer instrument that allows us to investigate a variety of metabolic pathways and which pathways our cell type is using and prefers to use to promote their own survival, growth, etc. And so it's becoming clear that different cell types can have very different preferences in what type of metabolic assays they prefer. And also a change in metabolic state can really indicate that there's some sort of issue or ill health of your cells. So changes in metabolic profiles can really indicate that you have some sort of difference in cell fate, or your cells are behaving differently than you would expect them to. So it's a really nice functional readout for analyzing stem cell behavior and fate decisions. And we also use a lot of live cell imaging instruments. So this here is an image from IncuSight Live Cell Imager that the Braid Lab has, and they generously let us use. And this is a really cool instrument where you can image your cells while they're in the incubator. The camera is in the incubator with them, and so you can take images as long as you like and sort of follow, morphological changes as your cells are developing. And what I'm showing you here is a neurite analysis tool that we've been using quite a bit to analyze differences in how neurons develop in the different genotypes that we're looking at. So with this tool, it actually, will take pictures over a time course, and then the system that's integrated into this instrument will identify individual cells. So it marks, traces the cell nucleus, that's in yellow. And then the pink markings are actually traces of neurites. So we can look at these as the neurons are first developing, see when they start to develop neurites, and then over time watch and measure how long they become, how interconnected they are with other cells. So for, when you're looking at doing some high throughput assays and eventually trying to perturb your cells with different compounds and environmental stressors, this is a really nice tool. And so I just want to take a minute to show you how high content imaging works at a basic level, because a lot of what I'll show you today is using this type of technology. So we have a Nikon TI2E microscope in our lab that has high content imaging software integrated into it, and we use that on an everyday basis. And we can look at, a couple hundred wells of cells at a time if we want to. But we also have here at SFU really incredible capacity to do really high throughput assays with this kind of approach. So at the Center for High Throughput Chemical Biology, you can do this looking at hundreds to thousands of wells of cells if you want. And the center has a library of over 100,000 compounds, bioactive compounds, natural products, drugs, some drugs that are actually clinically approved that you can use to screen your cells for changes in cell phenotypes. And so when we look at this type of data, what's actually happening, there are many platforms and systems that are set up to analyze cells, your images in this way to gather quantitative information. And this is a platform that Lisa in the lab has developed through Cell Profiler. And basically what is happening in analyzing this data is that the machine will extract your images. It will use a nuclear dye to identify single cells. So it traces those nuclei, and now you have a coordinate for every single cell that's in your well. And then it will, you can feed into it anything else that you have stained your cells for in that dish. So here, this is an organelle. I think this is mitochondria. And the system will then go and look at each of these cells that's identified and expand the trace that it gives you based on what the organelle staining looks like. And so now you have, single cell data, and you do this for multiple images that will give you quantitative information about every organelle or protein or whatever you choose to look at. So really powerful technique. Okay, so today what we're going to talk about is the research strategy that my lab uses. I just took you through that. And then the meat of the talk, I'm going to take you through an example of a rare disease that we've built stem cell-based models for. The disease is tuberous sclerosis. So it's an epilepsy and autism disorder that arises during embryonic development. And this is work that I started as a postdoc. So this is the rare disease model that kind of got me into this field in the first place. But we're still working on it in my lab. There's lots of big questions around this disease that we can attack now with the models that we've developed. So we've got some really cool projects that are expanding from this work. So I'll show you how we built this model. There's sort of three main cell types that we wanted to model that are found in the focal malformations that these patients develop that cause epilepsy and some of the other features of this disease. And I'll show you how we uncovered organelle signaling processes in a fundamental cell stress response that we think really underlies the phenotypes in this disease that we're now starting to understand. And then I'll just take a few minutes to show you what some of our current and very near future directions are. Okay. So TSC is, it's not just a brain disease, actually it's a multi-system disorder. What I mean by that is there are multiple organs and tissues in the body that are affected by this in this disease. And it's all due to one mutation. I'll take you through that in a minute. And all of these tissue types are affected. But really one of the main clinical manifestations that causes the most severe phenotypes for these patients is in the brain. And that's, as a neuroscience lab, what we're mostly focused on. But what happens is you have growth in all of these tissues of just malformations called hamartomas. So these are, also called low-grade tumors. They're basically malformations of cells that are just really abnormal. They're cells that shouldn't be there, or they're similar to the cells in that tissue, but they've just sort of undergone a really aberrant differentiation process. They're a little bit mixed up. And they form these sort of focal tissue defects. And so I'm just going to take you through the main manifestations that arise in the brain. So there are two main types of malformations that appear in the brains of these patients, cortical tubers and subependymal giant cell astrocytomas. Basically, these contain the same type of abnormal cells. They mostly differ just in the location that they're found in the brain. So these subependymal tumors obviously are in the ependymal region within the ventricle underneath the ventricular zone where the stem cells are. And cortical tubers are distributed, these sort of regions of focal malformations distributed throughout the cortex. Some patients will have, one or two, other patients will have multiple tubers that are hard to keep track of. So a wide variety in presentation in these patients. But nevertheless, the vast majority of TS patients are epileptic. That's really heterogeneous as well. Some will have, seizures here and there, but other patients are having multiple seizures a day. So it can be a very debilitating disease. Many patients are also on the autism spectrum or have some degree of intellectual disability, and it's largely thought to arise from these focal malformations. So in the field, it's actually thought that, it's quite clear from what we already know that these CNS lesions arise from a neural stem cell that has this mutation that's really abnormal. So it's neural stem cell driven. The other tissues in the body that are affected, the leading hypothesis is that this comes from a mesenchymal stem cell. So we call it CNS versus mesenchymal aspects of this disease. I'm not going to talk a lot about these, but just touch on it a bit because we found some nice comparisons between these mesenchymal cell types and the neural model that we've built. So I'm just going to point out one of these mesenchymal aspects of this disorder called lymphangioliomyomatosis, or LAM. This is a really awful disease that affects, it's largely unique to women with TSC, and it causes progressive lung destruction. And this is because there's these really strange population of smooth muscle cells that arise and grow, slowly grow, but over time continue to expand in the lung. So you can see here, this is the normal lung, just an H&E stain, and then from a LAM patient. And these are smooth muscle cells. This is the fluorescent image. Smooth muscle cells are in green. Normally, those cells are only around a blood vessel, but in LAM patients, they're all through their lung parenchyma, and these create cysts, excrete enzymes that degrade the lung tissue over time. And eventually, within a decade or two, leads to respiratory failure. So really diverse disease, but again, all due to one mutation in a single gene. And it's a relatively rare disease. And there have been some improvements in treatment options for these patients, but outside of surgical resection, which is sometimes possible, there's really one pharmaceutical intervention that is available to these patients, and that's treatment with rapamycin. That's a potent inhibitor of the mTOR pathway. Talk about this in a minute. That's a critical pathway in this disease. And it has, it helps a lot with different aspects of this disorder. It can inhibit epilepsy to some degree in many patients, but it's not a cure. So a lot of patients don't respond at all, or they become refractory over time. Side effects are really severe. So there's really a push in the field to understand the roots of this disease better, what's driving disease in each of these tissues, and to look at this in a human cell population, so that we can really learn what we should be targeting to sort of treat these patients. So as I alluded to, this disease is driven by dysregulation of the mTOR signaling pathway. Now, this pathway really functions in all of your cells. Just one piece of biochemistry for the talk. But it's really important. It's a critical homeostatic regulator of cell energy needs in your cell. So it balances and makes sure that the cell is producing or taking in enough energy depending on its needs. So what's really important for cell function is that you're able to properly regulate this pathway, activate it when you need to, inhibit it when you need to. And in TSC patients, they have mutations in one of two genes, either TSC1 or TSC2. And these genes, they make proteins, hamartin and tuberin, that function to make a complex together that inhibits activation of the mTOR signaling node. So it really comes down to disruption of this regulation point. So the way this pathway works, when there's lots of energy, ATP, growth factors, and nutrients in the cell, the TSC complex is inhibited, the mTOR pathway is on, so it can use all of that energy and material to promote anabolic activities that fuel cell growth, proliferation, lets them live happily. And when the mTOR pathway is active, sort of the main thing that you see is an increase in protein production. So this is a really important pathway for protein translation. And it activates other organelle systems like lipid and mitochondrial synthesis, drives metabolic pathways. When the cell has a low amount of energy and nutrients, the TSC complex is turned on so that mTOR is inhibited. You're not sort of using the little bit of material you have in the cell to fuel these activities, but instead you activate these catabolic processes, autophagy and lysosome biogenesis, to break down whatever is available in the cell to try to create new substrates to fuel cell growth again. So you can imagine when you have a mutation in one of these two genes, you cannot inhibit this pathway properly ever. So it's on a constant overdrive hyperactive state. So as I mentioned, we know this pathway is really important in many cells and is emerging as quite important during brain development. And many members of the pathway have been implicated in brain diseases outside of TSC, so other cortical malformations, different epilepsy syndromes, autism as well. And so this is, a bit of a better view of how complicated this pathway is. Just to sort of drive home that point that it's really interconnected. And so we think, this could be why the rapamycin, which directly inhibits mTOR, is not enough to treat these patients long-term, because these cells can, adapt to other pathways and find a way around it. So we really need to understand what other vulnerabilities there are in these cells to figure out how to properly treat the dysfunction that they cause. Okay, so at this point, we set out our stem cell lab and thought that we could sort of make some inroads here, and decided to create a human stem cell model to understand how the aberrant cell types in the brains of these patients develop. And we we started from a point where the field had really learned a lot about the biochemistry, really, that I just took you through, from fly and rodent models, immortalized cell lines. And some labs are starting to be able to get access to human tissue samples from embryos that had this condition and were aborted. And we've started to understand, again, that these cortical tubers and all the cell types in them likely come from a stem cell that has a TSC mutation early on, and that goes through different decisions to create all of these cell types. But we really wanted to investigate this now in a human cell model. So this is a general approach that we took to do this. So we started with pluripotent cells. See them in cartoon cells in the dish here. And instead of using patient-derived cells, what we did here was we used CRISPR-Cas9 to engineer a mutation in TSC2 that would make sure that the protein wasn't produced. And we did that because we wanted to create a really robust modeling system where we have the same mutation in 4 different cell lines. We have 2 male and 2 female lines, which is kind of important to represent both sexes if you can in this type of work. So we created those cells. Once we had them, we validated that there's no TSC2 protein produced. We can differentiate those into neural stem cells simply by changing the factors in the media that they're growing in. And the really important thing here is to supply the cells with 2 small molecules that inhibit the TGF-beta and BMP signaling pathways. And that really just ensures that we're recapitulating pathways that are activated or inhibited during actual embryonic development. So just mimicking what we know about development already. And within 12 days, we have a largely pure population of newly formed neural stem cells. So we also generated a model to look at mesenchymal cells and how they develop in the presence of these mutations. And I like to talk about this because I think it's a really cool developmental system that we can use to create these cells. So has anyone here, have you guys heard of the neural crest cell population? Okay, so we kind of exploited those cells. And another hypothesis in the field is that the mesenchymal manifestations of this disorder might actually come from the neural crest. And this, one of the reasons why is that we know neural crest cells and neural stem cells during early development are specified in parallel with one another. So as soon as the neural tube closes, and at this point it's completely full of neural stem cells, we have this population of neural crest cells that is specified between the neural tube and the surface ectoderm. It's really early in development. And they quickly acquire migratory characteristics, and they move outwards and seed all of these different developing tissues. So it makes sense as a hypothesis and was really handy for us because we could use the same media, almost the same approach to generate mesenchymal neural crest cells as we did to make these neural stem cells. The difference is simply sort of a mechanical one, where instead of differentiating these cells in this media on a dish, we actually start them as a free-floating cluster of cells. And when you induce them into neural stem cells in that way, it actually creates sort of something that reflects the neural tube, because it's this sort of self-assembled free-floating cluster. And if we plate them on a dish, sort of midway through this process, you actually observe neural crest cells that get specified, and they delaminate from this neural stem cell cluster and move around the dish. So you can either fix the cells with the neural stem cells and the neural crest cells together and look at that, or you can just pop off that neural stem cell cluster with a pipette, and you're left with this dish of neural crest cells. So that's how we made the neural stem cells and the mesenchymal neural crest cells in our model. And so I just want to highlight this feature of the disease, the bit of the genetics, so that you understand, what's happening in patients and what we're actually modeling. So this disorder, again, it's due to a mutation in TSC1 or TSC2 most commonly. The model we've made, this mutation is in both alleles of this gene. So all of the cells have, mutation in both alleles and have really severe phenotypes. But what actually happens in patients is they inherit one mutation on one allele in the germline. And it's only as the tissue develops, you'll have some cells that will randomly acquire a mutation in the other allele for TSC2. And it's only those cells and the progeny they make that will express the phenotypes that I'm going to show you. So the model we made, again, it has both alleles are mutated so that we could really look at these cells in isolation and understand what's driving their dysfunction. So just really quickly, going to brush through the slide. I just want to make the point that we were successful in the neural crest cell model that we made. We produced these cells from wild type cells and TSC2 deficient cells over 12 days, and then looked at some phenotypes to see if they could reflect some of the key features of lamb cells, and we were successful in that. So they make the right neural crest cell markers, they're more mobile, high levels of mTOR signaling, and they expressed two markers that are actually diagnostic for these patients, so that was pretty important in success of this approach. So again, I'm just showing you this because I'm going to talk in a couple points to show a comparison between what we learned about neural crest development versus neural stem cell development. So on to the brain modeling. This is sort of what we're trying to create in the dish. So again, there's two types of tumors these patients develop. And I want to point out to you again that, here on the left of this cartoon, this is where neural stem cells usually reside and persist in the ventricular zone right around the ventricle, and they stay there. And of course, you have your neurons and glial cells that populate the cortical tissue. But in the cortical tubers of these patients, they have a mixture of really enlarged neurons and glial cells and these stem-like cells that accumulate multiple different phenotypes that I'll take you through. But the point is that there are stem cells that are present in the cortical tissue, which is very strange. So when you look at tissue biopsies, here on the left is from a normal brain tissue, and on the right is a cortical tuber that was dissected from a patient. Just to H&E staining, you can see that the cells are really abnormal, quite large, completely disorganized. They've totally lost the normal architecture that cortical tissue usually has. You see high levels of mTORC1 signaling. This marker, PS6, this is a phosphorylated ribosomal S6 protein. It's a protein that's really important for increasing protein translation and is usually activated right downstream of mTOR pathway. So this is a marker we typically use to show activation of that pathway. High levels of glial cells, and that is something we've seen very clear in patient samples. And the neurons that are there are really large, dysplastic. They have long, thick neurites. And we have this population of stem-like cells that the field has coined giant cells because they get really large. They express high levels of stem cell markers like PAX6 and SOX2. And so it's really these three cell types that we're trying to make in the dish. And I'll tell you that so a few other labs in the field have used pluripotent cells to make neural and glial cultures to try to really understand the neuronal activity and dysfunction there. And that's where most of the field has focused. And we incorporated that into our model, but we really wanted to see if we could reflect or build a model that would reflect the development of these giant-like stem cells that are also found in these tubers. So we reasoned then that these cells are all coming from a neural stem cell that lacks TSC2, and that, they can differentiate directly into neurons and glial cells, and that these giant cells, a lot of evidence in the field to suggest that it takes a bit of time for these phenotypes to develop, so that it might just be, as the cells mature, they start to develop various aberrant phenotypes, which include, these increased stem cell markers, and increased expression of a number of organelles, and large cell size and these really large vesicles. So we devise a strategy where we create these stem cells, and then we just passage them repeatedly in a dish. So passaging just means you, every week, you take the cells off the dish, put them onto a new one so they can keep growing, and that lets them sort of mature and age in culture. So our strategy then is in the first two passages, we call this an early stage of giant cell development. The next few passages is a mid stage, and after that, we call it a late stage. And I'll show you how we have been able to observe through progression of some really key disease phenotypes using this approach. So the most obvious phenotype we first looked for was expression of stem cell markers. And by using this strategy, we do see increased expression of SOX2, nestin, GFAP, key stem cell markers that increases over time. So we're really happy that we could actually visualize progression of this phenotype. They have high levels of mTOR signaling, of course, and much larger than their wild type counterparts. And so in my lab, we started looking at these a little more carefully at the sort of mid and later stages of their progression, and have been pretty shocked to see these really large vesicles that sort of develop in our cultures. And then, went back and realized that there's evidence of this in the literature, where some cells in biopsies from cortical tubers of these patients have these giant vesicles. And there's one mouse model that was able to show this as well, but it took many months of postnatal development to see this phenotype. So we think this is sort of a mid to late stage progression of these cells. Wish I could tell you what they are. We've tried staining them for various things. First thought that they would be really large lysosomes. They're not. We've done some electron microscopy, and it's shown us that they have no membrane and they've got some protein in them. Some of them are empty. We don't know what they're for, but it matches phenotypes that we see in these patients. You can also pretty clearly see that there is a high level of organelle signaling, especially lysosomes in green and mitochondria. And this is something that we can use high content imaging for, and we see that this also progresses over time. So we're pretty satisfied that we have a nice model that's allowing us to follow the progression of these giant cells in a way that we in the field haven't been able to before. So finally, because of all of these organelle-based changes, we're really interested in understanding how this affects their metabolism, and if we can use that as sort of a nice functional feature of cell dysfunction, and what sort of phenotypes we're dealing with. And so I'm just quickly going to orient you to how the Seahorse analyzer works and is able to give us a readout of metabolic activity. So this instrument, basically you plate your cells we use a 96-well dish. You put it into the instrument. This plate has a sensor on it, and once it's in there, the instrument puts this cap on it that has these sensor probes. So these probes are able to detect the amount of protons in the media surrounding your cells, as well as the amount of oxygen. So I'm really focusing here on oxygen consumption. And that's a readout of mitochondrial metabolic activity, because that pathway is really dependent on consuming oxygen to proceed through the electron transport chain. So here, this plot, if you look at the x-axis, there's a number of measurements. So it takes measurements over time. These are usually about 3 to 5 minutes apart. So it can track how much your cells are consuming oxygen and how quickly. So here on the y-axis, this is the oxygen consumption rate that the instrument has revealed to us. And so I'm not going to take you through all of the details of how this is done, but each of these points is just an activator or an inhibitor of the electron transport chain, so we can get sort of different information as to how the cells are using this pathway. The take-home message is that TSC2 deficient neural stem cells have a much higher oxygen consumption, so a much higher mitochondrial metabolic rate than do our wild-type normal cells. This is like really a strong phenotype. Haven't seen many cells that are this highly metabolically active. And so it has a higher basal respiration rate. And then also this sort of peak here is telling you what its maximal respiration rate is. So, this drug here is sort of perturbed function of the electron transport chain, and it's forcing the cell to do as much as it can. So this tells us that these cells are really resilient, and they've adapted some adaptations that allow them to continue to respire and to stay alive in the face of different stresses. And so we can see, so here's passage 6 and then passage 9 beside them. So our mid and late stage model, where respiration rate does decline over time, and that really fits with the fact that we see the cells are proliferating much more slowly, but we still have this increased capacity in the knockout cells. So just a quick note to say that this is quite different than what we see in other types of TSC2 deficient cells. The neural crest cells we made and cells that other people have looked at, typically they have, a higher glycolytic rate, but not so much in the mitochondrial pathway. Other differences we see are, of course, we have an increased amount of lysosome signaling, which is counter to what the real biochemistry of this pathway is. So most cells, that pathway is shut down. So there are some really core things here that are telling us that neural cells respond very differently than other cell types to a loss of this protein and to mTOR pathway dysregulation. So we also differentiated these cells into neural and glial cells. And the main thing here is by providing them factors like brain and glial-derived neurotrophic factor, and we can get mixed cultures of neural glial cells in about 6 to 8 weeks. But a cool note is that TSC2 deficient cells actually develop, differentiate, and become electrically active neurons pretty quickly. So, within about 3 to 4 weeks, it's just quite fast for IPS neuronal models. Okay, so we found a lot of phenotypes that we know are present in patient tumors that reflect the fact that these neurons with this mutation have a much larger soma, these white blobs here, it's just a fluorescent stain, much larger than their wild type counterparts, and they have many more neurites extending from them. They're longer and more connected. We can see this increased capacity for glial differentiation. So this has also been seen in patient samples, that there's more glial cells at the expense of neural differentiation. And when I was in Ottawa working on this, I had a great collaborator at Carleton University, Hongyu Sun, who performed patch clamp experiments on these cells and found that they have a much higher amplitude and frequency of spontaneous hPSCs that can be restored by treatment with rapamycin. So we know this is this hyperactivity is because these cells can't properly inhibit this pathway. So in my lab, we started looking at this using some more high content tools as we're working towards doing those types of experiments. And are able to see with this IncuSite instrument, we can recapitulate this sort of lengthier and more interconnected neurites. And this is work that Melinda and Christina in the lab are doing. Lisa in the lab is working on developing calcium imaging tools. And already we can see that using this probe, it's just a live cell dye that actually gives you a readout of calcium activity. And we can see our TSC2 deficient neurons really quickly start to become electrically active. So all of this data that we had collected and others in the field had seen similar things kind of suggests that, maybe TSC2 deficient cells differentiate into neurons and glial cells quite readily. But, we realized that we kind of don't really look at these cells until they're, a month or two along, and there might be some different things happening earlier on. So we started looking at, here, this image here is just 1 week after we've asked these stem cells to differentiate into neurons and glial cells. We've been quite surprised. There's some other evidence sort of popping up in the field too, that this is a little bit more complex. So you can see at this stage, wild type cells, they're already developing neurites. So they're starting to become immature neurons, but in our knockout stem cell cultures, they're not. So their morphology is still that of a stem cell. We're still, we're sort of just getting into this and trying to figure out what state these cells are actually at, but a big difference. And, you can see the wild type cells as well has started to express these sodium potassium channels that show up early in development. And we don't see that typical pattern in the knockout cells. So this is actually suggesting that, maybe these TSC2 deficient stem cells are resistant to differentiation at first. Once they develop into neurons, they produce long neurites and they get really hyperactive, but maybe all of these cells don't want to turn into neurons. And so, we took that information and kind of modified our strategy to differentiate these cells. And we've gotten this to a point where we can actually visualize in a dish. This is a very early stage of differentiation, that there are stem cells that choose to remain as stem cells and some that do differentiate. And it's really cool with these microscopy tools that we have. We can measure this and sort of get a quantitative readout of how many stem cells are still in the population and how many cells have differentiated. And when we let them grow for a month or two, we actually see this really interesting self-organization. This is work that Lisa in the lab is doing. So it's been really fun to see, how these cultures develop. And they seem to sort of self-organize into these clusters of stem cells. And we get these neurons that organize themselves around the periphery. And so, we're wondering if it's somewhat reminiscent of how these actually develop in the brain. So we're going to use the system to start to find things that can find regulators of these fate decisions. And hopefully, can use this as a tool to measure when we get a change in the ratio of these cell types. And then we can track over time how this changes the formation of these structures that we see. Okay, so this is suggesting that we see a lot of organelle changes, functional changes in these giant cells and the neurons and glial cells they give rise to. But again, it's this sort of a common issue that the field is facing is which of these things do we target and what's really fundamentally underlying the dysfunction in these cells. So we thought to go back to our protocol to induce these cells at very early stage of neural lineage induction, where they're first exiting from pluripotency and turning into neural stem cells for the first time. So we did this, again, I'll show you a little bit with the neural crest protocol and the cells that we made here in parallel with the neural stem cells that we generate. So we took over this 12 days, we took samples of cells at different time points and did a variety of different analyses with them at these different stages. So the first thing I'm showing you is just a Western blot looking at activation of the mTOR pathway, but it's pretty informative. So again, we've used phospho-S6 protein as the main readout of mTOR activity. And so here on the left, these are wild type cells that are differentiating into neural stem cells and our TSC2 deficient cells. So day 0 is actually when the cells are still pluripotent. So you can see that the mTOR pathway is actually really high in those cells. But what was first striking to us is that in wild type cells, when they're induced into the neural lineage, they immediately shut down this pathway. It's a really big difference. But clearly the TSC2 deficient cells can't do that completely. And this activity persists for some time. And so our thought is that, wild type cells really need to shut this pathway down to develop properly. And so this must really cause a problem for these cells, that they can't regulate this pathway. And so I'm going to bring your attention again to this sort of co-culture approach that I described, where we can look at cells. So here we fix cells that still have that neural stem cell cluster attached to the dish and the neural crest cells outside of them. So we can just look at these two populations at the same time. And you can see that the neural stem cells really shut down this pathway, but neural crest cells don't. Likewise, neural stem cells have this accumulation of aggregated proteins that we don't see in the neural crest population. So I'm telling you this because basically, in the next couple of slides, I'll show you, we saw some really big changes biologically in our cell population that are turning into neural stem cells, but we really saw very little in this neural crest population. So it's quite striking because, they're really close together the way that they're induced, but they have very different responses to this pathway. Okay, so, one of the big experiments we did with these cells that taught us a lot was a series of RNA sequencing experiments. So we took samples of these cells, inducing into the neural lineage every 2 or 3 days. And we found, at day 0, this is pluripotent cells. So actually, what I'm plotting here is genes that we found or transcripts that we found are upregulated or downregulated in the knockout cells compared to wild type cells that are differentiating at the same time. So in day 0, these are pluripotent cells. There were, I think, 4 genes that were differentially expressed. So that really tells us that loss of TSC2 really doesn't affect pluripotent cells at all. But once you induce these into the neural lineage, we saw thousands of genes that were differentially expressed just in the neural lineage. So we took those genes that were upregulated and put them through a gene ontology tool, and that just takes the genes that are in your list and tells you what kind of cellular processes they're associated with. And what we found was a very quick response that activated an endoplasmic reticulum stress and an unfolded protein response. So that's telling us that there's lots of unfolded protein in those cells, and they've mounted a stress response to deal with that. And then later on, we see an increase in the amount of lysosomes, other cellular vesicles, and autophagy signaling. And this is just showing you these are genes associated with lysosome biogenesis. Again, it's just the neural stem cells, but not the neural crest cells that mounted this response. So we can validate this with live cell imaging and show an increase in aggregated proteins, lysosomes, lipids, other organelles in our TSC2 deficient cells compared to the wild type cells that are becoming neural stem cells. 2 interesting things to point out here. In these red boxes, the sort of 2 boxes or columns beside it are those same cells treated with rapamycin, and that restores the increase in these phenotypes. So again, that tells us that all of this is because the cells differentiating into the neural lineage cannot downregulate mTOR signaling, and that is causing a lot of problems. By the endpoint of this differentiation, these phenotypes actually all restore. So this seems to be a really strong but transient response that gets the cells to a healthy state, allows them to differentiate, and then the system's shut down. But as I already showed you, when we let these cells continue to develop, we see a lot of these same phenotypes reappear. Right, and so this is just another reminder that normally we see if the mTOR pathway is hyperactive, you should have an inhibition of the lysosome autophagy signaling, but we see the opposite in these cells. So what is this telling us? Suggesting to us that cells that are going to become neural stem cells, it's really important that they downregulate mTOR signaling, but in cells with TSC2 mutations, they can't. And this mounts a very rapid endoplasmic reticulum stress. And that's because there is an accumulation of proteins that's probably not folded properly. The ER is responsible for folding proteins. And so when this happens, the unfolded protein response is a really common response that cells will make. So there are multiple things going on with this response. There's 3 main arms of the pathway, but they each activate genes that, they have a lot of different functions. So they'll also activate organelle signaling and metabolic changes, proteins that are chaperone proteins that help those proteins fold and help to take them to the proteasome to hopefully, get them degraded. And it also will activate lysosome and autophagy signaling, as well as proteins to create more proteasome activity in an effort. So both of these systems can help to clear out aggregated proteins. So cells will mount this, and sometimes the response is sufficient enough to clear out the problem in the cell and to allow the cells to thrive and to continue to survive. But sometimes the damage is too much and the cells will die instead, and they make that choice through this signaling mechanism. So we think, again, that neural stem cells lacking this gene activate this response because they have this accumulation of protein. And we think that this likely is to make sure the cells stay alive, but it has downstream consequences. So Shama in the lab is starting to look at a protein level, which arms of this pathway are actually driving these phenotypes so that we can start to manipulate it in a more defined way. And she's finding that although we saw, genes involved in all aspects of this pathway that were upregulated in our cells, the protein level, there are changes throughout, but it's really hinging on activation of this IRE1 arm of the pathway, which is nice because then we can sort of mutate that gene and get at the mechanism in a really specific way. But what's also cool about this is that this pathway is linked to upregulation of the proteasome, so that structure in the cell that will really help to degrade the proteins that are accumulating. So we think that's probably a really important part of this response. Okay, so this is telling us that this cell stress response is really underlying the basic early development of these cells. And so now we're starting to ask, is this response really what is driving the phenotypes in these cells as they, develop into neurons, into these giant cells? Is this what's really driving the dysfunction in this disease? So we went back, this is just a couple of slides of data we're just starting to collect in our giant cell progression model. And this is some really cool data that Lisa is starting to collect. So she's working out how to profile our cells, the organelle staining patterns in our cells for various organelles that we have seen are implicated in this cell stress phenotype that we found, and linking that with cell metabolism to try to understand, which parts of everything that we're seeing are really responsible for some of these functional changes that we see in these cells. So up here, this is on this side is wild type cells, and these are our TSC2 knockout cells. Again, they're neural stem cells, passage 3 or 4, and marked with a live cell probe for the ER in green, mitochondria in red, lysosomes in yellow, and again, nuclei in blue, so you can identify single cells. So clearly, there's a very different pattern in our TSC2 knockout cells compared to wild type cells. They look very different. And I've shown you this before. The oxygen consumption in our knockout cells is in red, and the wild types are in black down here. Huge difference. But what's cool is when we treat them with rapamycin, we see, the wild type cells don't look that different, but a bit of a diminishment of some of these visual phenotypes in our knockout cells. And metabolically, it brings it right down. So, okay, clearly, still we're looking at something that is a consequence of this hyperactive mTOR signaling, even at this stage. And what was also really interesting is when she treated these cells with, this is stapsagargan, so one of 2 compounds that we're using that activate an ER stress. So we can look at this, outside of just our mutation, and activate it in different ways. So we can see that this really changes the organelle pattern of our wild type cells. And at the metabolic level, here is wild type respiration in black. And activating cell stress in these wild type cells brings that metabolic rate way up to where our TSC2 knockout cells are. So this is some nice data that's further suggesting to us that cell stress signaling is actually really contributing to the metabolic and other changes that we see in these cells as they're sort of progressing towards a giant cell and likely making these abnormal neurons. Okay, and so one quick snippet of data that I did a while ago, but really hammers this point home. So if we treat these cells with agents that inhibit the proteasome, that sort of structure that can help degrade proteins, we get a really high killing of our TSC2 deficient neural stem cells. That's in red and yellow. Wild type cells are largely fine, and we don't see this effect in the neural crest cells. So again, really pointing to this cell stress response and this dependency on activating, organelles like the proteasome to keep these cells alive. Okay, so in summary, showing you that I've been able to model tuberous sclerosis, 3 key cell types in this disease that are linked to epilepsy and autism dysfunction in these patients. Built a model to compare neural stem cells and mesenchymal cells, how they develop differently in the context of this mutation. And that's starting to tell us maybe about how we might need to target different aspects of this disease for a treatment paradigm in a different way. And we've also uncovered this cell stress lysosome signaling response that we think is really underlying the survival of these cells, but probably a lot of the phenotypes that we see as well that are causing all of this dysfunction. Okay, and so really quickly, just going to flip through where we're going next. So while doing these experiments, we've also found a lot of data points that suggest there is really a change in cell fate and cell identity in these models that's correlating with a lot of the stress and organelle phenotypes that we're seeing. So I showed you already, as these cells progress in our giant cell model, they activate over time, higher levels of stem cell markers. We see higher levels of these markers like PAX6 during that early development phase that correlates to some degree with an increased expression of certain organelles like lipids. And we can also see when we make cerebral organoids, so these 3D brain tissues from these cells, that we have an increase in lysosomes, again, in the knockout tissue, but it's completely confined to the stem cell zone. So suggesting that there's really a link there with function of the stem cell population. And Shama in the lab has started to look at this and really trying to understand how that early cell stress might be affecting cell fate decisions over the long term. So this is a really cool experiment that I just wanted to show you because it's such a striking result. So what she did here was when these cells are pluripotent, the first day that we ask them to turn into neural stem cells in the neural lineage, she treats them with stress-inducing compounds, and then takes it away after a day, lets them continue to develop for these 12 days, and then differentiates them, passages them, here are 3 or 4 passages. So this is a good 5 or 6 weeks after they've been exposed to this stress insult. And here's what the cells look like when she looks for markers of neurons and stem cells after all of this time. So wild type cells, they're mostly PAX6 positive stem cells. These are in media that are meant to support stem cell expansion. But our wild type cells that are treated with that stress agent look very similar to our TSC2 deficient cells at this time point, where there's a mixture of stem cells, but others that are starting to express beta tubulin. They don't look quite like neurons yet, but they're starting to activate this marker that might suggest that they want to differentiate. And then these double stress cells, so our knockout cells that have been treated with thapsigargan, there's very few stem cells left, and they're mostly neuronal, and they very quickly stop proliferating. It's quite a profound result. So this is an avenue that we're really excited to keep exploring. I mentioned this model, and we're going to start doing high throughput compound screens to try to identify compounds, and these are linked with cellular processes, right, that will change the ratio of stem cells to neuronal differentiation. And these are some of the new directions that we're taking and are really excited to build over time. So through collaborations, largely here, we're starting to model 2 new rare diseases using the same kind of approach I told you. So spinocerebellar ataxia is one of them. And this is a project in collaboration with Randy, where our lab will do the stem cell modeling parts, and then we aim to merge that with the brain imaging AI work that he does through the virtual brain to try to inform, with our models, give a deeper understanding of the neuronal dysfunction in these patients. We're also developing collaboration with David Vicadlo's lab to model Batten disease, and with stem cell technologies to use the compound screening center to figure out how we can better produce dopaminergic neurons from iPS cells. So we're also really interested in trying to improve our ability to make human cells in these models. So along with that, we're also embarking on 3D bioprinting. So we have a 3D printer in our lab. If anyone is interested in that, let me know. We'd like to get more users on it. And Shama has started work on that, printing our normal stem cells, our TSC2 deficient cells, in hopes that we can create a more 3D model of these cortical tuber cells, and seeing some interesting results with that already. Okay, so very important slide. I'd like to acknowledge my wonderful lab. Most of them are here, and some of our friends in the Braid lab as well. I think it's time for a new picture with everybody in it. Yeah, and that too. So you can actually see what they look like. So a lot of people I mentioned throughout the talk, but there are people here that did great work in the lab that I just didn't have time to talk about their work today. But this is a great group of people that I'm really thankful for. Some of the wonderful folks in the Stanford lab that I worked with at the start of this project, and collaborators in Ottawa and here. And of course, our funding sources relevant to this work. Okay, so thank you very much for your attention.